Welcome back to another episode of the Mushroom Show. Here at FreshCap, we appreciate mushrooms of all shapes and sizes, but perhaps we can pay a little more attention to the root structures of mushrooms, otherwise known as mycelium. Today's episode is all about these mycelial networks and the incredibly important role that they play in our ecosystems. And we are honored to have Dr. Toby Kiers here to enlighten us on the subject of underground fungal networks. Dr. Toby Kiers is the co-founder and executive director of SPUN, which stands for the Society for the Protection of Underground Networks. Their mission is an ambitious one. It is to map all of these mycorrhizal fungal networks across the world and then to advocate for their protection. The team at SPUN believes that these vast fungal networks, which in general we probably don't think about very often, are actually integral to clean air and clean water and the general healthiness of our planet. In our conversation, Toby helps us connect the dots between plants and fungi, the important roles again that these fungi play in the forests around the world. We talk about their role in carbon sequestration and also the incredible work that Toby and her team have been doing in Patagonia and the work that they plan on doing in other really cool areas in the world, where Toby and her team are looking for biodiversity hotspots to really study and understand, again, the roles that fungi play in our world. I think you're going to find this conversation truly fascinating, and I really hope it's going to be enlightening. So without further ado, here's our conversation with Dr. Toby Kiers. Dr. Toby Kiers, welcome to the Mushroom Show. I am super excited to have you here and super excited to be talking to you about underground networks.
Yeah, it's such a pleasure to be here. I'm excited to talk about these mycorrhizal fungi and systems underground that are totally underexplored.
I totally agree. I think they're totally underexplored, and this topic, I think, is something that is also totally underexplored in terms of how much people know about these amazing networks. But I think where I wanted to start is, how did you get interested in this in the first place? How did you get interested in fungi? Because mushrooms is kind of a niche topic, but underground networks specifically is a niche topic, but it does have huge implications. So why don't we just start by talking a little bit about your background and how you got into this?
So I think I started to think about soils before I started to think about fungi. And I like soils because they have this sort of thrift, like they don't give up their secrets very easily. And fungi are like that too. They're this very mysterious kingdom, and it kind of takes a bit of an elaborate courtship to actually coax them in the lab and to be able to study them. And so I think that challenge has always been super exciting. But I really started my work on fungal networks when I was 19, and that was in the tropical rainforests of Panama on this little island called Barro Colorado Island. And this was like the late 1990s, almost 2000. And we really didn't have any idea what was going on with fungi underground, but it was such a hotbed for tropical research. So many scientists have come to this one island to study all kinds of the above ground biodiversity. But because we were there and all of the scientists were focused on things above ground, I was really curious what was happening underground. And I guess that's kind of where it all started, was looking at these immensely diverse tropical forests and just thinking, my God, what does it look like underground? Is it just as diverse below ground as it is above ground?
Well, that's a really interesting outlook and a really interesting way to think about things. And it's not super intuitive either, right? Because I think, too, even if people are interested in mushrooms, they might be interested in harvesting the mushrooms or picking the mushroom out of the ground and not really thinking about all the stuff that's happening all of the time underneath our feet. So maybe just briefly explain to the listeners, what is a fungal network anyways, and what makes them so significant in the natural world?
Yeah, so we use the word fungal network, but that's really shorthand for saying mycorrhizal fungal network. And we specifically study these mycorrhizae that form symbiosis with plant roots. So there's really two major types of what we call mycorrhizal symbionts. And the first is called arbuscular mycorrhizal fungi. And these associate with around 60 to 80% of all vascular plants. So really ubiquitous, right? If you were to go out in any kind of ecosystem and dig up a plant, there is a very, very high likelihood that that plant would be colonized by these fungi. And these are the fungi that really helped the first land plants colonize land some 450 million years ago. So plants used to live in aquatic environments, but they partnered with these fungi, the ancestors of these fungi, to actually start colonize terrestrial land. And the fungi were really good at breaking up rocks and turning them into nutrients and feeding them to the first land plants. And in return, land plants were feeding sugars and fats to these fungi. And so that's really what drove the above ground biodiversity that we see today, is that partnership that happened so many hundreds of millions of years ago. And now the symbiosis it's quite an exquisite dance between these two partners, because with arbuscular mycorrhizal fungi, they grow out of little spores underground, and they colonize root plants by actually penetrating through the plant cell, the root cell, and then forming a structure called an arbuscule, which actually looks like a mini tree inside a root cell. It's a beautiful structure that looks like really like sort of a tiny shrub inside an actual cell. And that's the site of nutrient transfer between the plant and the fungi. And so these fungi, they grow out into the soil and they collect phosphorus and nitrogen, and they trade it for carbon from the host plants, so in the forms of sugars and fats. So that's the symbiosis that we study today. And that's the kind of arbuscular that really we focus on in the lab. And the other main type is called ectomycorrhizal fungi. And those you might be more familiar with, because those are some of the mushroom forming species. Not all ectomycorrhizae form mushrooms, but a lot of your favorite chanterelles and truffles and things like that. So in that symbiosis, they don't actually penetrate into the cell, but form this herring net around the outside of the root system. But again, both partnerships are really dependent on the plant feeding carbon to a growing fungal network underground.
That's amazing. And these things can obviously be quite complex, right? You mentioned some of the species like the ectomycorrhizal fungi, like chanterelles and porcinis. Those are always mushrooms that I hear people want to learn how to grow. And it's like, well, it's a little more complicated than that, because these underground fungal networks and these symbiotic relationships are actually quite complicated, which you're studying. But one of the things that I wanted to try and understand is, oftentimes you hear about these mycorrhizal relationships as being in symbiosis. And people think this is kind of like, the plants and the mushrooms are holding hands and they're just very happy with each other and they're living in perfect harmony. But it is a little more competitive than that, right? It is a little more of a kind of a capitalist environment. Why don't you explain a little bit more about like how the interactions between the fungi and the plants are, and the things that they're trading, and some of the strategies that mushrooms use in these kind of competitive capitalist relationships?
Yeah, well, I think using the word capitalism is it's a loaded word, to say the least. But we definitely know that they participate in these underground economies, and they're very simple economies, right? And I think that it makes sense. We see a lot of these economies emerge between lots of different cooperative partnerships in nature, and the mycorrhizal symbiosis is one of the sort of most dynamic and exciting to study in terms of these trade, because what we see is that these fungi are incredible at evolving these strategies that really allow them to optimize their interaction with plant roots. So what I mean by that is that a fungal network, these arbuscular mycorrhizal networks, they're colonizing multiple plants simultaneously. So that means that they're forming these complex networks that tap into multiple plants simultaneously. At the same time, they're out there foraging for nutrients like phosphorus and nitrogen. And in our group, we've been able to label these nutrients with these very tiny particles called quantum dots. They're these nanoparticles that fluoresce in bright, pure colors when exposed to a UV source. And what that does, it allows us to track nutrient movement across these networks at an unprecedented scale. And so we can actually see the fungi engaging in these strategies where they're able to optimize the way that the nutrients move to really increase their gains from plants. So let me give you an example. A fungi might take up resources from a spot where there's lots of nutrients available, let's say to both the fungi and the host root, actively move it across its network, and then trade it with a plant root where demand is higher. And by doing that, it actually gets paid more. And so this is incredible. Not only are they able to sort of calculate the nutrient richness in different patches, but they can also sense this temporal element. So what they can do is they take up nutrients and then they actually hoard them in their network. They keep the phosphorus in this form that's unavailable to plants called polyphosphate. They hold it really tight in their network until phosphorus depletes outside the network, and then they feed it to the plant. And again, they're getting more carbon payoff by holding it back. And so time and time again, we sort of set up these experiments where we're trying to understand, okay, how are they calculating? How are they actually able to process the information about where resources are high and low? And time again, they surprise us with these very, very clever strategies.
And I think that's so amazing. This is an organism that's performing these actions and making decisions almost as if it has a brain, when it clearly, it doesn't, it's not like a human where it actually has a brain as a control center. It's more of just a distributed network of intelligence somehow. And there was another example of a study that came out a few months ago where people were hypothesizing that mushrooms have a language of up to about 50 words. And when we take that kind of thing and combine it with what you were talking about there, with mushrooms somehow making decisions about where to transfer nutrients to get the most value, it kind of gives you this thought that maybe these fungi are somehow a lot more intelligent than we give them credit for. Is there any explanation, or what is the most clear and obvious explanation of how this actually works? Or do you think it's just kind of natural forces within the mycelium that are doing this?
Yeah, again, I think this word intelligence is loaded, and so we have to be very careful. I mean, we know that fungi have no central nervous system, right? They're not animals; they're fungi. But like other organisms that are exposed to natural selection for hundreds of millions of years, they have to be able to navigate their environment, and they have to be able to obtain resources and trade those resources in ways that increase their reproduction. And so again, they're able to sense resource gradients. That's the same as, let's say, a plant growing towards a light, or even a root system growing towards nutrients. And so it's built into their structure. Their structure is very complex, but that idea of being able to move towards resources and having gene networks that allow them to sense those resource gradients is quite accepted. And so again, to turn that into words like intelligence, that's where the big step happens. And I think that happened with this language paper as well. So what we see is that fungi have calcium channels, they have ion channels, just like most animal and plant cells do, and those generate electrical activity. And so it's really interesting to try to understand those pulses and try to understand what those pulses mean. um, Again, there's a lot of, let's say, what we call signal to noise ratio when you're looking at things like electrical activity. Does that mean that it's a language? I think we really have to be rigorous in trying to understand what these types of electrical pulses mean and how they would help the fungal strategy, whether it's growth or moving nutrients. And I think that's when we'll really get at the heart of what's going on with fungal information processing. There's no doubt that they process information, right? All organisms are able to process information, but how they do that across these complex bodies is a really important question. As soon as we bring in words like language and intelligence, it's a little bit murky, the water. So I'm just trying to stress that it's built into their genome how they move across the landscape, and it's just us who don't understand how it works.
That's a really great point, and I'm glad you add a little bit of clarity to that, because it is super important to understand. Because these networks are important, and we talked about how they're trading resources and how they're kind of integral to the environment in the forest. But fungi are also sequestering carbon in a way, which I think is really interesting. I think that's something that people might be surprised to hear. So can you explain a little bit about the importance of fungal networks in terms of sequestering carbon and the important role that plays in our environment?
Yeah, so this is really exciting. I think that the world is sort of waking up to the importance of these underground systems in carbon sequestration. And this really happens through three main routes, I would say. So we know that there's CO2 in the atmosphere, and then plants photosynthesize, and they turn that CO2 into photosynthate, which is sugars. Now, what they do with that carbon is allocate it below ground in the form, as I said, of sugars and fats to these growing fungal networks. So they're basically, because the fungi need that carbon to grow. So I didn't mention this before, but these arbuscular mycorrhizal fungi, they're what we call obligate biotrophs. So that means that they depend 100% on plants to complete their reproductive cycle. They cannot survive in the absence of plant roots. So they're feeding this carbon. They actually act like a bit of a vacuum, sucking the carbon below ground and putting it into their expanding network. Once the carbon is below ground, it's used in three ways. The first is to support, to fuel the cells as they grow with that big network. The second is that these networks, they create what are called exudates, right? These are these sort of secondary compounds that act to keep the aggregates of the soil sort of glued together. And that's the third thing that these fungi do. So they're creating exudates, which means more carbon underground, but that more carbon underground is sticky. These fungi produce substances that make the soil sticky so that aggregates don't wash away. They're really important in stitching ecosystems together. And so by doing that, they are drawing carbon below ground. So about 75% of all terrestrial carbon is stored below ground. So even if you added up all the plants and animals, it's still not as much carbon is stored below ground. And so again, these systems, we have to kind of think of them as like the circulatory system of the earth, because they're the ones that are drawing the carbon down and keeping it down.
Yeah, I love there's a quote, I don't know exactly who said it, but I think I saw it on your website. It was, if trees are the respiratory system of the planet, fungi are the circulatory system of the planet. And I think that is a really great way for people to kind of internalize the importance of that. And the way you talk about these underground networks and fungi and the roles that they're playing and the importance of that, it seems like the world without fungal networks would be a pretty dismal place, and probably pretty soon a totally inhabitable place, and we should probably protect these networks. Which brings me to obviously the important work that you're doing. Now, you're the co-founder of SPUN, which stands for the Society of the Protection for Underground Networks. Why don't we talk a little bit about that and the work that you're doing in order to actually protect these fungal networks, and maybe highlight again the importance of why these networks should be protected.
Yeah, great. Well, thanks for bringing up SPUN. So SPUN is a nonprofit science initiative that is aimed at mapping the mycorrhizal networks of planet Earth, right? So we have basically three missions. We want to map all of the mycorrhizal networks, really try to understand the biodiversity patterns underground and how these networks stitch together ecosystems. And then we want to protect them, right? We really are engaged in advocating for the protection of underground ecosystems, through with governments and activists and researchers, so we can kind of shift the perspective from above ground ecosystems, a little bit more focus on below ground ecosystems. And the third thing that we want to do is really innovate around underground ecosystems, right? I think that if we put even just a fraction of the amount of resources that we put into going into outer space, to going to the underground ecosystems that we know so little about, that we'd really be able to harness these ecosystems to help with climate change. So that's why we founded SPUN. But right now, what we're concentrating on is those first two, is really trying to map all the mycorrhizal networks of planet Earth and sort of, advocate for their protection.
Now, it sounds like a simple task, right? Oh, let's just map all the networks on planet Earth, all the fungal networks on planet Earth. But I mean, that is an absolutely massive undertaking, and it's something that's never really been done before, as far as I could understand. Why don't you talk about how you did that? I mean, you mentioned quantum dot technology, maybe explaining a little bit more about what that is and how that helps you actually create these maps of underground fungal networks.
Yeah, well, first, it's funny that you say it sounds like an easy topic, an easy challenge.
Right.
Because every day I wake up and say, what did we say we were going to do? Map the Earth's fungal networks? No, it feels ambitious to us. But it's been wonderful. We just launched in November, so we're a brand new NGO, but we've gotten lots of support from it feels like from a lot of diverse voices that feel that these underground ecosystems have really been a global blind spot in conservation and climate agendas. So it's really fun to bring all these people together to start raising awareness about the soil. But in terms of mapping them, so when we say mapping them, what we're talking about is mapping the biodiversity patterns. And so what we do is we use observations that have been taken over the last 5 years or so, where scientists have actually gone out, taken soil samples, and extracted the DNA from those soils, and posted, through peer-reviewed papers, those sequences in a database. In this case, the database is called Global Fungi. And so we have about 10,000 of these samples. And what we do then is we put these sequences into these machine learning algorithms. And what those algorithms do is they help us predict where we think biodiversity hotspots underground exist. So it's basically using these, not only the DNA sequences, but looking at all kinds of climate variables and how different climates are to predict where we think these really unique niches are for biodiversity hotspots. So what this allows us to do is sort of predict where we think these really diverse mycorrhizal networks exist, and then try to go out there and sample and ground truth those predictions, because of course when you're using things like machine learning, they're just models. And so what you really have to do is then go out and ground truth these data to figure out what mycorrhizal species are actually there. So that's what we're doing right now with local communities, and this huge network of researchers are going out and actually taking these samples in a very standardized protocol and extracting the DNA and looking at the sequences and trying to understand what fungi live where, what mycorrhizal fungi live where, and what are they doing? What are the ecosystem attributes that they're associated with? We know that some of these mycorrhizal networks are really good at carbon sequestration, but others are very good at tight nutrient cycling. Some can survive drought conditions, whereas others can survive floods, right? We have to look at the world very pragmatically and think, okay, it's changing fast. And so we really want to get out there and understand what networks are where, what they're doing, and then fight for their protection.
So you just got back from a trip to Patagonia, which I assume is one of these biodiversity hotspots, and that's probably the reason why you went there to study that. What did you learn from that particular trip to Patagonia, or are you still parsing through all of that data?
We are still parsing through. So right now, those DNA sequences are at a sequencing facility in Valvida, Chile, where our collaborator César Morin, who worked with us to gather these samples. We learned a lot. There's a lot to do, right? As soon as you go out into the field, it's a different game. So you can sit behind your computer and you can look at these predictive, biodiversity maps and say, okay, we'll take about, 20 samples from there and 50 samples from there, and we'll be able to evaluate the biodiversity on, let's say, 75 pixels of Earth, right? If we call a pixel of Earth a 1 kilometer by 1 kilometer pixel, we can start putting these Chilean fungi, mycorrhizal fungi, on a map. It's not that easy, right? So this was an amazing expedition that was together with the Fungi Foundation. So Juliana Forci, which I think your listeners might know, and Merlin Sheldrake, who wrote the book Entangled Lives, and his brother Cosmo Sheldrake. And we all went out together to start ground truthing these predictions. And it was much more difficult than we had imagined. So, for example, our algorithm would tell us to go to a very specific location that was a predicted biodiversity hotspot, and it showed that there would be a road to get there. You'd get there and it was a swing bridge that was about to collapse, for example. So the conditions were difficult. It was hard to get as many samples as we had hoped, because it's one thing to pick up these hot spots behind your computer, but then when you're actually out in nature, it's a totally different ballgame. But we saw some of the most beautiful and diverse forests that I have ever seen in my life. So we were near the Lanin volcano, which is full of these araucaria trees, really beautiful ancient lineage of conifers that your listeners might know as the monkey puzzle trees. They kind of look like Lego trees, right? They're the very strange trees with branches that look like they evolved during the dinosaurs, and they did, right? They're really there's all these hypotheses that their leaves are so prickly and hard to ward off herbivory from dinosaurs, and you kind of feel like that when you're there. How ancient these forests are. Um, and they have really unique fungal networks. So they have what has been hypothesized is what are called these fungal nodules. So these kind of little balls of fungi on the root systems of Araucaria, which is very different. And usually these conifers like pines or spruces would be associated with Ectomycorrhizae. But when you go down to Chile, it's opposite, and these conifers are associated with these arbuscular mycorrhizal fungi. So that was really exciting to see that you can read all of these papers and sort of make generalizations, but once you're in the field, it's a totally different game.
Yeah, that is funny to think about. I mean, I see this a lot in like mycoremediation, where you say, oh, you can use mycelium as like a water filter, so why don't we just build these big mycelial nets that like, you can funnel rivers through and clean the water. And it's like, well, it's pretty easy to do that in the lab, but when you actually go out in the field, you run into issues like, I don't know, the bridge is blown out or whatever, you explained it. But at the same time, you get out in nature and it probably reinvigorates your desire to study and protect these networks for the benefit of the earth. So you went to Patagonia. Do you have like an agenda of where you want to go to next to continue mapping? Do you have kind of a hit list of these biodiversity hotspots?
Yes, predicted biodiversity hotspots, right? I mean, I guess that's what I keep trying to stress, is that if we really want to take underground conservation seriously, we need to build very rigorous maps of where these networks are, and actually figure out where are the endemic networks, where are the ones that are in trouble, where are the ones that we have to go out and save right now, and how can we drive adoption of, let's say, the first conservation easement just based on fungal biodiversity alone. We've got conservation easements for bird species and plant species, but not for fungi yet, right? And so to be able to do that and really make a difference, I think in conservation, we need to know where the networks are, what mycorrhizal networks are where, and this allowed us, this trip initially allowed us to do that. So Patagonia was a beautiful spot. It has decades of traditions, if not centuries, of people very interested in fungi there. So not only the Mapuche cultures that have been working with these fungal systems for so long, but also a really strong history of mycologists that have been taking surveys of mushroom forming species for decades. And so that's a great place for us to go, because now we can really compare what's happening below ground to these mushroom forming species above ground. So again, yeah, Patagonia is one place to definitely keep an eye on for even more future work. But the upcoming expeditions, we've got one going that's working with local collaborators in Ecuador. And here this is really focused on looking at how smallholder farms can use fungal networks for regenerative ag. So we're really interested in trying to understand the identity of these mycorrhizal fungi in working farms. And Ecuador is a really interesting place because not only is it a biodiversity hotspot for so many different kinds of plants and animals, it's also predicted to host very high fungal biodiversity. In these farms, they're usually close to forest ecosystems. And so what we're going to do is we're going to go and sample again with local collaborators in the forest edge, but then also in these farmer fields, and then in very high intense conventional agriculture, and try to understand the different filters that are leading to this decrease in fungal biodiversity. So what kind of fungi are in farmers fields depending on management strategies, and are those fungi associated with particular benefits in terms of stopping nutrient leaching. We know that fungal networks can decrease phosphorus leaching by up to 50%, right? They're really good at holding nutrients in the soil so they don't get lost into the waterways, and providing all of these benefits to farmers that I don't think are widely acknowledged. These networks are really good at increasing the nutrient density of crops. They make flowers bigger and nectar sweeter. So they're really important for plant pollinator interactions with bees, for example. We know that these networks can change the secondary compounds of plants, actually upregulate their defenses so they're better protected against herbivores and pathogens. But what fungi are doing that, and are they under threat? So again, focus not only in these pristine environments like these forests in Ecuador, but then seeing what is the spillover and how are farmers using these native fungal networks in cropping systems. So Ecuador is a big place. And then there's going to be an upcoming trip in November to a very far away place called Palmyra Atoll, which is in the South Pacific. It's one of the furthest places from any continent on Earth. So it's
Wow.
very hard to get to. It's owned by the Nature Conservancy. But there's really interesting nutrient dynamics, nutrient cycling between the coral reefs and the very nutrient limited rainforests on these islands. And so we're trying to understand the role of mycorrhizal networks in what's called forest to reef cycling of nutrients. And so that's a very exciting project. And yeah, on our website we sort of list some of the upcoming, biodiversity hotspots that we're very interested in. I think what we really have to concentrate on is, okay, so we can make these predictions, but what we really know is where samples have been taken in the past. And so we're working on what's called an underexplored map right now that basically looks at, okay, well, where are the places that samples have been taken before? What are the climates and habitats where fungal networks have been sampled before? So grasslands versus deserts, let's say. And if you overlay these two variables on top of each other, you get this map that glows very brightly in places that either haven't been sampled, that are a far geographical distance from the last previous sample, or a habitat that hasn't been sampled. And here, sub-Saharan Africa just lights up so brightly. I think there's really a lot of work that needs to be done with local collaborators in some of these sub-Saharan African countries where mycorrhizal networks have really been underexplored.
Well, it definitely seems like there's no shortage of places where you could go and study and learn more about fungal networks. And maybe the challenge when you have such a large goal of mapping all the networks is just figuring out, how to make it smaller and where to go to next.
Yeah, and just to add on to that, I think one of the things that we're really stressing in SPUN is that it's not about us going to these places. Every once in a while I get to go on an expedition, but really it's about working with local communities, local researchers, and providing them with the resources that they need to actually be able to sample soils in their own habitats, and use that knowledge and use those resources to build these open source maps. And so it's really important in today's world not to do what's called helicopter science, right? Where people from developing countries are going to the global south and just sampling, and that's it, right? So really, what we're trying to do at SPUN is to work with local researchers and communities to help facilitate sampling on the ground.
That makes a ton of sense. And again, so you said that, first step is mapping, the next one is protecting. But what are some of the threats that are being faced by these fungal networks? Why do they need to be protected, and aren't they just fine if we just let them be?
Yeah, if we could just let them be, they probably would be fine. I mean, they've survived through what, 3 or 4 mass extinctions already on Earth. Their fungi are amazingly resilient. The problem is the speed at which climate change is happening now and the intensity of land use changes. So I think things like agricultural expansion are probably one of the most threatening um, global change drivers to fungal networks. So these fungi are really important in agricultural systems, but when you have really fast and intense agricultural expansion using conventional methods that are heavy in fungicide, fungicide kill fungi, and nutrient inputs, then that can be really dire to these mycorrhizal networks. So we know that plants feed fungal networks to get at nutrients. And so if you just keep adding lots of nutrients to soil systems, plants stop feeding these fungi, right? So that's really can lead to their disappearance. We also know that tillage is incredibly, dangerous for the fungal network. Um, there's data suggesting that in tillage systems, it reduces these network forming fungi by about 2/3. So, and it selects for these very fast sporulating fungi that don't form the sort of nets that you need to protect against a nutrient leaching. Roundup, common chemicals like Roundup, have been shown to decrease the diversity of spores, or the viability of spores. I think it's the viability of spores. Um, Up to something like 700%, right? So Roundup can be very dangerous for these spores. Logging, right? I mean, I could just keep going on. It's really depressing statistics, but logging, yeah. I mean, you're basically ripping out the trees that support these fungal networks, and that can reduce the abundance by 95%. Like it's really intense. These logging systems, what they do to underground ecosystems. Um, Fire is bad, not for all fungi. Thank goodness there are some that love, when a fire comes through, but not mycorrhizal fungi, right? They find it very hard, and it can take decades for these really diverse communities to come back. So we're worried, right? I mean, the UN is saying that by 2050, about 90% of the Earth's soils are going to show some signs of degradation. In some places, we've lost all our topsoil completely. So I think the urgency is something we feel right now. And if we can focus on below ground conservation and kind of have this shift, this mind shift, if you will, towards conserving things we can't see, then we're going to be in a much better state to fight climate change.
That's really cool. And I guess it's a tougher sell, right? And I guess that's why it's important, the work that you're doing, because if people can't see it, it's like, well, like who cares? Like it's almost like if you don't see it, it doesn't bother you. But if you can show and map these networks and show how important they actually are, and maybe help, people on a bigger scale form some sort of relationship with these underground networks, then that will probably help these protection and conservation efforts.
Yeah, Tony, I think that's a really good point. Like, I just think that, we live in this really visual world. And so, of course, we want to protect the things that we can see. So one of our job at SPUN is really to how, to try to ask, how can we tap into other senses that allow people to experience soil systems in different ways? So, for example, in my lab here in Amsterdam, we work with biophysicists at the Amolf Institute to actually visualize the nutrient flows inside these networks. And once you see what a moving, living, nutrient river of a fungal network looks like, it kind of changes your whole idea of what soil is. Like the whole earth kind of shifts beneath your feet. It feels liquid all of a sudden because, these networks can make up 50% of the living biomass of soils, and they are constantly moving. And you've got these two streams that are moving simultaneously in different directions, right? You've got carbon moving one way and nutrients like phosphorus and nitrogen moving the other. And these fungal networks, they're not septated, right? So most fungi have septa, and that's these cells walls that break up the cells into compartments. But these mycorrhizal fungi are like an open pipe system, and you can just see these really complex flows inside a living, evolving network. It really shifts your perspective. You just see how complex it is, and again, that's trying to get at fungal behavior and trying to understand how fungal behavior can change the way that carbon is sequestered underground, because it's the behavior that is allowing this carbon to move across these massive networks in ecosystems. So again, focusing on little, even at the micron level, trying to understand fungal behavior and how these carbon flows are controlled by fungi, and what stimulates them to move the carbon faster or into different places, are really important for these underground systems. So visually, I think that's important. We can study the flows. We can see how a fungi is calculating by moving these nutrient streams in different directions. and it totally changes the way you look at the underground. The other exciting thing I think is in our trip to Patagonia, we were with Cosmo Sheldrake, who was using these very sensitive microphones to record sounds of the underground. And wow, I mean,
Yeah.
it's really dramatic. It sounds very different.
Yeah.
Um, and different places sound different. A grassland sounds different than under one of these araucaria trees. It's just, we're starting to understand that these are called soundscapes, and the ecology of soundscapes, and using acoustic signals to look for biodiversity hotspots. That's what they do above ground with birds and insects and things, but how can we use those technologies below ground? I think that's a really exciting research frontier. And it also allows people to connect with those sounds, to hear things. So yeah, I think using different senses and really trying to strengthen that connection between people and soil systems is going to be massively important going forward.
Yeah, and I'm glad you mentioned that the sound. I was listening to that this morning, and it's such a weird sound. It's so unique and so interesting, and it kind of reminds me of there's other people on YouTube that are like putting electrodes onto fruiting bodies of mushrooms, and then like running them through a synthesizer and hearing the sound. And I think, yeah, the more we can like connect to these things, the more we'll realize their importance in some way. And I think too, like, you probably notice a big influx of interest in mushrooms in general over the last little while. Stuff like fantastic fungi, for example, which didn't specifically focus on underground networks or mycelium, but it definitely talked about that. And I think it changed the perception in people's mind that mushrooms aren't just these things that grow on dead logs that we don't care about. Mushrooms are actually integral into our entire environment. And I think more people that realize that and get interested in mushrooms in general, I think is going to be a very much so net positive for the planet as a whole. I don't know if you've noticed kind of the same thing.
Yeah, definitely. I feel like, that the awareness of the fungal kingdom is growing and growing fast, but it just can't grow fast enough, right? And so I think if people want to get involved, I think something that they can do right now that could be very helpful, is sign the 3Fs initiative, which is flora, fauna, funga, which is advocating for people recognizing the kingdom of fungi alongside with flora and fauna. So instead of just looking at diversity of flora and fauna, that we really advocate for the inclusion of funga in treaties and in governmental documents and in museums. And as soon as our lexicon starts to adopt things like words like funga, the 3Fs, it can help change perspectives and have people concentrate on this kingdom that has been historically so underfunded and overlooked.
Right, but and I guess it leaves a big opportunity for people to study and learn, and yeah, I mean, it's a never-ending. You could never learn anything about fungi. Like there's like we have definitely not done enough research, and even though there's lots of people studying these things for a long period of time, there's still so much more to learn. And I do love the idea. I mean, you mentioned people signing the 3Fs initiative, but I do also love this idea of like citizen science or people who are just getting interested in it. Maybe they're not scientists or maybe not studying it in the capacity that you are, but what are some of the other things that just everyday people can do if they want to kind of help push this initiative along?
Yeah, I think that's a really good question. We ask ourselves that a lot every day, So how do we get people activated? Um, so at the SPUN website, we're calling on what we call myconauts, right? This is a great word, myco meaning fungus and not meaning explorer, you know, to really help us activate the larger community. And so, within that community, we do things like go to climate protests, and try to advocate for underground ecosystems, which is fun. I mean, even at climate protests, people are like, "Wait, what are you trying to do?" It's kind of a new, it's kind of a new look. Like we're trying to protect underground ecosystems, right, for conservation and for climate mitigation. And people get excited because it's sort of a new way of looking at action and what they can do to help. So yeah, I mean, eventually what we're aiming to build is this whole sort of Myconaut network that allows people, citizen scientists, community members to be able to sample in their own habitats, in their own backyards, and be able to have that be sent to a local DNA sequencing facility, so that we can start really understanding these sequences in a broader pattern at a much higher resolution. That said, it's still really hard. It's a long way off. Citizen science works really well with things like counting mushrooms, and things like counting birds, and things that people can go out and see. It's really hard for citizen scientists to go out and do things like these arbuscular mycorrhizal networks, but we're getting there, right? I mean, I think especially with the emergence of things like DAOs, like these decentralized autonomous organizations, I think the way that we do science is shifting massively, and I think that citizens and community members are going to become so much more involved in the future of science, much more than we can predict right now. It's a heavy, clunky machine right now, right? It's hard to do the kinds of sequencing that we need to look at the genetic makeup of these. Is expensive and clunky, and so that's really why we're trying to build a pipeline that makes that a little bit more efficient, so that everybody can be involved. There's really exciting DNA sequencing technologies emerging that maybe people don't know about, things like nanopore techniques that basically allow people to do more sequencing in the field, right? And the quality is increasing every day in terms of how good the sequences coming out of those approaches is. So I can see that within a couple years we're all going to be able to sort of sample what's in our own habitats and have a better understanding, not only of who's there, but their function, right? I mean, I don't know how much you follow the news, but like we're also making huge progress in trying to understand sort of what we call like the gene regulation, the what's called metatranscriptomes, like the sort of the gene pathways that are upregulated or deregulated under different conditions, so that in real time you can basically see function, right? And that's really exciting. It's still a little bit science fiction in terms of doing it in your own backyard, but I think that science moves pretty fast, and pretty soon it's going to be more equitable, in the sense that we'll all be able to sort of look into the systems that interest us and understand these systems that are right below our feet.
I love the term Myconaut, and actually I tweeted the other day, I said, born too late to explore the Earth, born too early to explore the universe, but born just in time to explore the Mycoverse. And I think that's what you're doing with SPUN. I think it's really cool. The timing is perfect to be doing this. So spun.earth is the website. Where could people go if they wanted to learn more about everything that we talked to today? Would the website be the best place?
Yeah, spun.earth would be the best place. And we really want people to sign up. And we're getting good about sending out newsletters and updating people in terms of upcoming expeditions, the ones we've been on, the ones that are being planned by local collaborators all over the world. So there's ways to help and to stay connected, I think, with this new underground climate and conservation movement. So spun.earth is a great place to go. If you're really interested in the nitty gritty of things like fungal behavior and flows and topology of networks and things, then you can go to my lab website, which is just my name, tobykiers.com. And that gives us all the peer-reviewed scientific papers where we're looking at fungal decision-making and strategies.
Excellent. We'll put the links to all of that in the show notes and the description of this video. Dr. Toby Kiers, thank you so much for joining us on the Mushroom Show. This is a really enlightening conversation, and I think, people are going to find a lot of value learning about all these fungal networks.
Thank you, Tony. I had a really nice time. This is a great show, and I wish you all the best of luck.
Awesome.